Myself Biswajit and I'm from IIT MANDI. And, we are sitting in lab like Center for Human-Computer Interaction, where most of the PhD researchers and postdoc students working on different types of related to human-computer interaction. We generally design low-cost products like heart rate variability, EEG, FNIRS, and early detection of Alzheimer's disease and other things. So my guide name is Shubhajit Chowdhury, and I am from IIT MANDI. I'm a PhD student. So today we'll discuss about the development of a pulse mode photobiomodulation for enhancement of cerebral oxygenation level. So organization talk, at first we'll talk about the introduction, then materials and methods, then circuit design, hardware design, study design, results, application, and then references. So I'm working on photobiomodulation. So at first we should know the meaning of photobiomodulation. The meaning of photobiomodulation is photo means light, bio means biological tissue, and modulation means alteration. At 1967, Henry Master, who is a scientist, once he working with tumor cell with lasers, then he found the cell grow, hair cell grow on tumor cell. So that is why he is well known as the father of photobiomodulation. The mechanism of photobiomodulation is that if we put light on cell, light penetrates into the cell of the mitochondria. In mitochondria, there is a cytochrome C oxidase, which has the ability to interact with lights. When light interacts with mitochondria activate and it produces energy form of ATP, adenosine triphosphate, and nitric oxide and reactive oxygen species, which helps to enhance the oxygen level. And photobiomodulation, previously known as a low-level laser light therapy, and the wavelengths used in case of photobiomodulation is between 600 to 1100 nanometer. So materials methods, in my circuit there is a two-part, hardware part and software part. In hardware part I use a power supply and a step-down transformer, then AC to DC converter circuit, then my PBM circuit, and NIRS means or you can say FNIRS means functional near infrared spectroscopy. So here you can see PBM module. So this is the architecture of my main module. So here there are total of 7 blocks you can see, and every block there are 4 circles. Red circle represents 660 nanometer LEDs, and yellow circle represents 850 nanometer LEDs. Black circle represent 810 nanometer LEDs. So blocks are divided between 2 rows. In first row there are 4 blocks, second row there are 3 rows, 3 blocks. So as vertical line, there are distance between 2 blocks is 1 centimeter, and horizontal line distance between 2 blocks is 2 centimeter, and diagonally distance between 2 LEDs is 3 centimeter, and vertically the difference distance between 2 LEDs is 2 centimeter. The overall height and length of the device is 5 centimeter and 14 centimeter. My device is also it can angle between 60 degree, and I designed my LEDs on a normal sponge. So I use 2 sponges, upper layer and lower layer of sponge. So the overall height length width of the device is 9, 19, and 3 centimeter. So here's my circuit design. So at first here is a 220 volt power supply, which is connected with step down transformer. And step down transformer helps to step down the voltage from 220 to 9 volt. And 9 volt then connect with AC to DC converter circuit, which I designed on breadboard. And here I used also IC 7805 voltage regulator, which helps to convert voltage 5 volt. And then power supply and 5 volt connect with my main PBM circuit. And here I use ESP32 microcontroller for control purpose, and it is connected with laptop for getting power supply. So there are the whole hardware design. So you can see number 1, 2, 3, 4, 5, 6, 7. So 1 is the single switch, then 2 is transformer, 3 is AC to DC converter circuit, 4 is ESP32, my controller, and 5 is my device. So here you can see the blink of red lights. So we can see red lights with eyes, but we cannot see the infrared ranges of light with our naked eyes. So and here 6, you can see the FNER devices. So FNER helps to monitor the present oxygen level of the head. Suppose our device, this is our device, and if we put our device in the frontal head, then light penetrate into the tissue of the head in interacts with blood chromophores, then it produce oxygen. And those oxygen we can measure with FNERs. So here's the software part is software part you can see in laptop, and FNERs we brought from I forget actually. Okay. Okay. So there is the study design. So study design divided between three parts: pre, during, and post. During P, we collect present oxygen level with the help of FNERs, which we placed in our FP2 area. This area is called FP2 in frontal head. And then we used our device named Photobiomodulation. So for 10 minutes, and then we collect post data with the help of FNERs again, and we compare post and pre data. So here is the graph, the pre and post. You can see that there is subject 1, subject 2, and subject 3. In every graph, you can see the post saturation is much more higher than pre. So there is a term STO2. STO2 means skeletal tissue oxygen saturation. So which is increased with but we can increase, it is increased by time. And there's a one more term is BVI, means blood volume index. Suppose photobiomodulation helps to enhance the oxygen level into present blood. So brain creates signals like we don't need more blood, because there is a concept like more blood means more oxygen. So when we don't need more blood means capillary going to narrow, and then blood little bit reduce. So here post signal going to reduce. And the application of photobiomodulation, generally photobiomodulation applies for different types of brain disorders like stress, anxiety, depression, stroke, traumatic brain injury, Alzheimer's, Parkinson, dementia, etc. Here are the references. Thank you.